Hello everyone, I'm Dr. Omidullah Faizi and I'm really glad to have you here. In today's lecture, we'll begin our discussion of chromatin structure, the foundation for understanding how DNA is organized within the cell nucleus. In this lecture, we'll explore what chromatin is made of and how it's packed. So let's begin our first topic, chromatin structure. Before we go deeper, I want you to imagine something. If we take the DNA from one human cell and stretch it out, it would be about 2 meters long. Yet, still, all of that DNA fits inside a nucleus that's only a few micrometers wide. How is that possible? The answer is chromatin. Let's see how chromatin makes this possible. So first, let's define what chromatin actually is. Chromatin is a complex of DNA and proteins. There are two main types of proteins found in chromatin: histone proteins and non-histone proteins. So, DNA, histone proteins, and non-histone proteins are the main components of chromatin. Now let's imagine this small segment here as a part of DNA molecule. DNA molecule itself is made up of three main components: a nitrogenous base, a deoxyribose sugar, and a phosphate group. The phosphate group carries a negative charge because it contains several oxygen atoms. This is why the entire DNA molecule is negatively charged. These phosphate groups connect one deoxyribose sugar to another, forming what we call sugar-phosphate backbone. This whole part is called sugar-phosphate backbone. Sugar-phosphate backbone is present at the peripheral parts of DNA molecule. These are sugar-phosphate backbones. Nitrogenous bases are present at the central part of DNA molecule. So, DNA is negatively charged because of its phosphate groups. Now here we have histones. This is DNA molecule. These are histone proteins. Histone proteins are small positively charged proteins. Because they are rich in amino acids lysine and arginine, due to these amino acids, histone proteins carry a positive charge. And negatively charged DNA wraps around these positively charged histone proteins to form chromatin fiber. Non-histone proteins are also present, but they play a different role, mainly in regulating gene expression and maintaining chromatin structure. Now, let's take a closer look at the smallest structural unit of chromatin, the nucleosome. This is a nucleosome. Just like a wall is built from bricks, chromatin is built from many small repetitive units called nucleosome. The nucleosome is the basic building block of chromatin. Each nucleosome helps fold and organize DNA in a very precise way. So, the DNA loops twice around this group of 8 histone proteins to form nucleus. These 8 histone proteins together are called histone octamer. Histone octamer. So remember, octa means 8, and that's why we call it a histone octamer. Now let's assume this is a histone octamer. This histone octamer contains 2 copies of each 4 core histones. H2A, H2B, H3 and H4. I've shown them in different colors. For example, blue ones are H2A, black ones are H2B, red ones H3 and green ones are H4. So now, as we mentioned before, the DNA wraps around histone octamer, making about 2 turns. Then a short stretch of the DNA connects one nucleosome to another. This part of the DNA is called the linker DNA. Linker DNA. So linker DNA connects one nucleosome to another. And the 5th histone called H1 binds to nucleosome and linker DNA. Now, it looks like beads on a string. Each beads represent a nucleosome, beads, nucleosome, and a string is the linker DNA. Linker DNA. So, the entire arrangement here is called beads on a string model. And it's about 10 nanometers in diameter fiber. So now that we understand the nucleosome, let's move one level higher. We'll see how these nucleosomes are further folded and compacted to form the structure we finally observe as a chromosome. Chromatin organization is not random. It's very systematic and hierarchical process. Now let's talk about the levels of DNA packaging. Levels of DNA packaging. DNA packaging occurs in several levels, from 10 nanometers in diameter fiber up to 1,400 chromosomes. Now, this is the level that we already discussed, what we call beads on a string model. Next, these nucleosomes are coiled further to form a thicker fiber about 30 nanometers in diameter. This level of folding making DNA nearly 40 times more compact in its extended form. These 30 nanometers fibers then form loops, which attach to scaffold protein inside the nucleus. Scaffold proteins. These loops are about 300 nanometers wide. These loops are further folded and compacted to form a 700 nanometers chromatin fiber. At this stage, the chromatin is fully condensed, which allows it to fit efficiently into nucleus during cell division. Finally, chromatin is folded even more to form metaphase chromosome. Which is about 1,400 nanometers wide. So from 10 nanometers fiber to fully condensed metaphase chromosome, DNA undergoes a series of systematic and hierarchical folding steps, making it nearly 10,000 times more compact than its extended length. Alright everyone, that's all for today's lecture. Today we learned about the structure of chromatin, from DNA and histones to nucleosome formation and higher order organization up to chromosomal level. In the next lecture, we'll discuss the types of chromatin. See you in the next lecture. Goodbye.